Hello, this is Dr. Clark, and today we're going to go into veterinary drug development and control. So what this is going to go over is the main stages and steps in actually developing a drug and safely bringing it to market. And a lot of what we're going to go over is on the veterinary side, but it has a lot to do with the human side. As far as the testing and stuff goes, it's not that much difference between the human and the veterinary side. It's just at the end of the day what the label says. So, in the United States, all new drugs have to go through a mandatory series of tests uh, through the FDA. And so the FDA says you have to complete all these different stages, all these different tests, and have to prove the safety of your drug, the efficacy of your drug. And if you do all this, then you can actually sell your drug on the market. So we're actually looking at developing new drugs. There's four major steps in drug development. So the first is synthesis and discovery of a new drug compound. And there's several different ways to do this. This could be through bacterial compounds. This could be through chemistry in the lab. A lot of times we'll find them from plant sources, or potentially even now we find different things from animal sources. And we will actually synthetically make that and actually try it in the test tube, or what they call in vitro in the lab, and see how that works. Then we actually have to go through safety and effectiveness evaluation. So we're actually going to see how safe this drug and how effective it is. They're going to start off usually by testing it in mice and rats in the lab, and then we actually test it in the species that's intended for. And then they actually submit a review of what they call a new animal drug application, or a NADA, NADA. And then that's what the FDA is actually going to sit there and look at. And then based off of that, they're going to make the decision whether or not to approve that drug. And then if the drug makes it to market, then there's what they call the post-marketing surveillance stage. This is where we actually come in and practice. So this is actually our observation of the drugs. Does it seem to be effective in our hands or not? Do we have a question about it? More importantly, are we seeing some sort of severe adverse reaction or increase of a specific adverse reaction that was not noted in the information given to us about the drug? And so those types of things we actually have the ability to report. And it's just not veterinarians that can report it. Owners can report it, and you, as licensed veterinary technicians, would be able to report it also. It usually takes an average of 7 years once the testing stage begins for a drug to actually hit the market. Actually, from the time of development to the time it hits the market, it's probably more than 7 years. But to actually go through all these evaluation and testing stages with the FDA and their final approval for a veterinary drug is about 7 years. And usually, for tests, the pre-stuff that they do is usually sometimes you'll have people that will spend 20 years working on one drug until they actually get it passed and all the way approved. So a lot of times your research and development part of this can be just as long, if not longer, than the actual approval process. So we kind of talked somewhat about the synthesis, safety, and submission and post-marketing. So when we actually move beyond that, we actually have four distinct stages that go on. And so in stage one of a drug, the scientists are actually going to synthesize and discover the substance, and they're going to do a bunch of preliminary studies. And these studies determine if the drug produces the intended effects and whether it has major toxic properties to it. And a lot of times when they do stage one, this is before the FDA is involved. So stage one is the very early stuff testing. And so, this is going to include computer modeling and seeing, now we can use computers to figure out chemically how things interact. We can have some idea how they interact in the body. We actually test in lab medias, like in test tubes or on plates. And then we can actually test on specifically bred bacteria and funguses to see how they work, especially if we're testing things like antibiotics to see exactly how they're going to work. But we can also get some biochemical tracers on other drugs by actually testing it on those. So that's our first thing. And then, we'll actually do cell cultures. So we'll take cell cultures of, say, liver tissue or of kidney tissue and see how this drug reacts with those. If it kills all the tissue on the plate, probably not something we're going to try in the animals. If it doesn't, then it's something that we can move on forward. So stage two is our preclinical studies, determine a drug's safety and effectiveness. And so in stage two, a lot of times you're going to actually figure out your targeted species, who they're going to test it for, and what they're going to actually do is they're going to be taking and actually working on things like lab rats and mice, and actually seeing how effective it is in them, and then looking at short-term and long-term tests. And so short-term, generally going to be less than 3 months. Long-term are going to be much longer than that. And so they're going to check for things like immediate drug reactions in the short-term test, any organ damage, reproductive effects, and then as we get into long-term, we also look at organ damage, reproductive effects, carcinogenity, which is causing cancer, teratogenity, causing any types of birth defects. So these are all things that they're going to look at in the initial preclinical trials on those mice. And a lot of times, like looking for carcinogen effects or birth defects or reproductive effects, one thing why we use mice is we can look at generations in a short period of time. And so they want to make sure that 5 or 6 generations down the road after you've used this drug, we don't start seeing untold consequences, that it's not done some type of damage to the DNA that would lead to a higher rate of cancer. So there's a lot of testing that goes on to make sure that we're not seeing an increase of cancer, birth defects, reproductive effects, and then in the animal itself that gets it, to make sure that we're not having any major organ damage. From here, we can submit what they call a new animal drug application, or an investigational new animal drug application to the FDA. And so once this is actually kind of put in and approved, then the clinical trials actually begin. I've been told that actually you have to do the new animal drug before you do the testing in the lab mice and rats. Books say something a little bit different. It could be either way, but before you actually do clinical trials, for sure you have to have this investigational new animal drug application turned to the FDA. And then you can actually start testing it in your targeted animals. And when you're testing it in the targeted animals before we get to stage 3, then what they're looking for is the same things that we were talking about before when we're doing this, is to make sure that when we're doing this, that we're testing in enough animals that we're not seeing any issues with the drug, that it's working appropriate in that species, we're not having any organ effects, we're not seeing any acute increases in cancer in those patients. And usually these trials have to go on for a period of time to where at least 2 or 3 years, to where we can test it clinically and see we're not seeing a reproductive effect. Or if we are, what type of reproductive effects? Are we actually having things like birth defects going on? Is it actually working effectively as it should? Are we having issues with liver, kidneys, or cardiac, or anything else, digestive issues? What are all the side effects and potential complications of the drugs? And so once we actually get through all this clinical testing and we make sure that it's safe and effective, then we can actually file what they call a new animal drug application, or that NADA. And so here all the research studies are submitted to the FDA. And then depending on what's being used, if it's in the drug and what the potential side effects are, there could be evaluations by the EPA to see how you would properly dispose of it, or the USDA to see if it's in food animal, how it would be in meat. If you're doing something like a pesticide, we do the same thing, except that the fact with the pesticide, you would go through the EPA, and when you would get to stage 3, then stage 3, they would consult with the FDA, make sure that there was no adverse reactions to the animals or anything. And then if everything's successful through this application process, and this takes about 2 years, where they're going to look at everything. So you might spend 7 years developing the drug, 5 years testing it. It can be 2 years to get it approved. And if they look at something in this stage 3 right here, when you do the application, that they don't like, they can make you go back and do further testing. So a lot of times, while the book says 7 years, there's a lot of times these drugs, from the time that we start actually testing them until we actually get them out to market, is 7 to 10 years because of this process right here. If they have any questions, you have to go back and prove or disprove. It cannot just be left hanging. So if there's something that's not clear in the data. Once everything's good, then the FDA will actually grant approval for that drug to be used. once everything's been cleared. And so then we get to stage 4 where we're involved, which is post-marketing surveillance stage. So this is where we actually get involved watching what's going on with the drug and seeing are there any adverse reactions going on that were not seen during testing. And so there's some very good examples of this. I know in equine medicine we had a drug named Baptin. I actually used it quite a bit, and it was used for tendon injuries. And these tendon injuries and horses would be crippling and debilitating, and they would not want to heal, and we would inject them with Baptin, and 2 months later they were healed and the horse was going back perfectly fine. And I myself used a lot of Baptin and really liked it and never had a problem. But when it was tested as a one-time use in horse. And so horses had started getting 3 or 4 injections for multiple tendon injuries. All at once we noticed this very high rate of anaphylaxis. And therefore the manufacturer pulled it off the market. We used to have the same thing with an injectable form of trimethoprim sulfa, which is available orally, but we had an injectable form for horses that went IV. And I've actually talked to a vet that actually did have, a colleague of his in the practice actually did have a horse that had a severe anaphylactic reaction. And when they tested it on 1,000 horses, it was a very low percentage of anaphylaxis. When we got out into practice with it, we found out that it was a much higher rate. So sometimes even though they do their best testing, there's times where we see untold consequences or things that did not come up in the testing for the way that the drug was intended. And so therefore, when we get into practice and we notice these things, it's our job to actually sit there and report them. And so this way, the FDA or the company might decide to go ahead and pull that drug off the market if there seems to be a problem with it. There's actually a couple of different numbers that you can call. One, you can actually call the drug manufacturer. And it's not a bad idea to do that. The only caution I'll give you is that they're commercially invested in this. And so not always sure if they're going to actually turn this information over to the FDA. There has been some problems on the human side of that actually not happening. So you can call the product manufacturer, and as a backup, you can also call the FDA at 1-888-FDA-VETS, V-E-T-S. So 1-888-FDA-VETS. And this number right here is the number that you call with any issues you have with any drugs, veterinary drugs. You can call that and they will record that and they will investigate it. Or you can actually call the United States Pharmacopoeia Veterinary Practitioners Reporting Program, which is actually administered by the drug companies lobbying group right there. But they actually record all these and they do turn all these over to the FDA for sure. And that number's in your book at 1-800-487-7776. Myself, I would probably call the USDA directly. But that's where we come involved in it is actually this post-marketing surveillance. Very important stage on any drug. When we're looking at safety and effectiveness, we're looking at short-term tests. So these are looking at hours following the test for adverse reactions. And also for anything that we see within usually the first 3 months. So anytime that we do these safety and effectiveness, we're going to be looking at the first 3 months in short-term tests. And this is where we usually start off as our short-term testing. Long-term tests usually go from 3 to 24 months with reported dosing, and we're going to usually be looking at various organ systems for toxic damage. So in this testing for toxic damage, this could be through blood work. It could be biopsies of like the liver and the kidneys. Some animals we will test it on and we will actually do autopsies and actually look at those targeted tissues and see what the effects of the drugs were. Most of the time we do less necropsies now and we do a lot more of biopsies and blood work to check out those organ systems. And pretty much long-term testing, 24 months is kind of the gold standard. Usually we're looking at 2 years and we're actually monitoring these animals out over a period of time with the long-term testing. And some animals might have a one-time dose in one group and then others will do repeated dosing just to see what's going on. Sometimes, and actually in many cases now, we're running a lot longer than 24 months on our long-term testing. We're looking probably closer to 36 months, and we're doing multiple groups on different ways of different dosing regimes. Specialized areas that we look at is reproductive effects, and so there's some things that aren't really needed to be reported. And so one is reproductive effects. Because it takes a large group of animals and you have to be breeding them to get past the reproductive effects. A lot of times you're going to read on labels not approved for reproducing animals. It's because they didn't have enough to actually prove its efficacy. So it's a lot better to actually put a cautionary statement and say, well, it's not for animals intended for reproduction. And so we actually do have that, or we have some that will say, not sure what the reproductive effect of the drug is. So we will test it, but sometimes it becomes unclear in testing exactly what contraception, fertilization, and pregnancy ends up being reproductively with some drugs. So that information right there sometimes is left a little bit vague on purpose. We do actually have to look at cancer-causing effects, the carcinogenicity of any drug. We actually have to sit there and go through and see what is the chance of this drug causing cancer. And if there's any chance whatsoever, it's not going to be used, especially for any food-producing animals. But if we see any significant increases in cancer, then that drug's not going to make it to market. Teratogenicity, this is actually causing birth defects. And so we're going to test this on pregnant animals, and we're going to do it at different stages of pregnancy. And we're going to see, okay, in this group, was there any known birth defects? And so once again, it depends on your group. There's a lot of things that's not approved for use in pregnant animals. They've done the basic testing. They didn't see any birth defects, but there wasn't enough numbers for them to say definitively there wouldn't be. So there's a lot of drugs that we actually do get that will say, not intended for use in pregnant animals because of that. Next, we kind of go to toxic evaluation. And so we start this off on mice. And we start off at a therapeutic dose, and we start increasing that dose until we actually see tissue or organ damage in the mice. And we determine what the highest dose is that permanently causes permanent injury or death to the mice. And so this is how we figure out, well, where is that toxic level for the mice going to be at? We also try to figure out where the effective dose is. So we have two different things that we have here called, one's called ED50, which is effective dose for 50% of the animals that receive it. So we figure out where, when we get to this level, let's say it's 1 milligram per kilogram, that this drug is effective for half of the animals that receive it. That's what we call the ED50 of a drug. There's also an LD50, and that's lethal dose 50%. So that same drug, let's say we bump it up to 50 milligrams per kilogram, and we find that half the animals we give it to ends up passing away. Then that would be the lethal dose 50%. And these are really important because we're going to get the safety margins next. And so when we look at the margin of safety of a drug, we're going to evaluate the effective dose 50% compared to the lethal dose 50%. Sometimes these numbers are way far apart. Things like Paranol, otherwise known as Strongyte that you all use, to deworm has a very high, unbelievably high safety margin, almost like 1,000. And then there's other things like we don't give ibuprofen to dogs because the effective dose 50% and the lethal dose 50% are almost the same number. And there's some animals you don't even have an effective dose, you just have a lethal dose like acetaminophen in cats. So all these are very important to know and understand. So when we look at the therapeutic index of a drug, this is the drug dosage of a drug that produces a desired effect with minimal or no signs of toxicity. Also called that margin of safety. And so to determine it, what we're actually going to do is take the lethal dose 50% and divide it by the effective dose 50%, and we're going to come up with a number. And so a wide therapeutic index means that a drug can produce the desired effect without actually damaging any tissue or actually killing the animal. So if we had a lethal dose 50% of, say, 100 in the milligrams per kilogram and a effective dose of 2 milligrams per kilogram, you divide 100 milligrams per kilogram divided by 2 milligrams per kilogram, gives you a therapeutic index of 50, which is a fairly safe drug. However, you take another drug and let's say its lethal dose 50% is 10 milligrams per kilogram, but the effective dose is 5 milligrams per kilogram. Well, you divide 10 by 5, you get a therapeutic index of 2. So we want a very high number on therapeutic index. And there's a lot of drugs like antibiotics that we give that can have that. NSAIDs can have that. There's other drugs that we have much more narrow ones, and you would expect that. Things like chemotherapeutic or even our anesthetic agents. So, it's very easy with anesthesia to overdose a patient and potentially cause harm or death. And so some drugs have wider therapeutic indexes we use, and we do have some with narrow ones. And most of those are going to be, like I said, chemotherapeutics or things that we end up using, such as anesthetics or some of our analgesic medications that are opioid based or narcotics. Additionally, when we start looking at it, additional testing, we do systems-oriented screening. This is where we are going to look at specific organ systems and see exactly how they're going to react to the drugs. And so we're going to take the GI system, we're going to take the nervous system, the cardiovascular system, and look at them. And then we're going to look at long-term effects. And then once again, we go back to our reproductive cancer and test for birth defects, and we're going to evaluate all these. And a lot of times we actually continue to screen after the drug is out in the market. There'll be continued testing by the companies to actually look to see if there's any untold effects that they didn't catch the first time. And then once again, it's kind of up to us to actually sit there and look at it and say, hey, we've been using this drug. It didn't say it, but we've been noticing that there's been an increase in kidney enzymes when we've started using this new drug. And this is something we haven't noticed before, and we've noticed it in this specific group. Or maybe it's the fact we've been using a drug therapeutically for, say, something like thyroid conditions, and we've had the animals on it for 5 or 6 years. And all at once now, we start actually seeing that all the patients we've had on it are now developing thyroid cancer. And so it's looking at things like this that's very important for us to do. And so, the company has an obligation to do that, but we also have an obligation to report that if we see anything. And so we're actually part of that chronic ongoing study of drugs. So there you have it. That is chapter 2, the chapter on drug safety and evaluation and development and control. So we will see you in lab.